now that we have a better understanding of what DNA is, the rest of Chapter 9 we're going to focus on looking at protein synthesis. And these are the objectives that will go along with that. So first off, protein synthesis. What is it? Well, it's the building of proteins. And so just a quick reminder from Unit 1, a protein is a large organic molecule. Remember, organic means it's got to have carbon and hydrogen in it. So it's a large organic molecule, and it's composed of amino acids. We talked about the fact that amino acids are the building blocks of proteins. Now, the reason that proteins are so important to be made inside of a cell is because each protein has a very specific function. These specific functions carry out pretty much every chemical reaction that goes on inside of your cells. If you remember, enzymes typically are protein in structure, and so proteins are very, very important. Now, the function of a protein is specifically determined by the shape of that protein. And the shape of that protein is determined as the amino acids are put together, the protein folds in a particular manner. And as it folds and takes on a shape, that determines its function. So all of this linked together in that process of building a protein. Now again, protein synthesis is the process of making that protein, but how do we know what protein to make? And how do we know how to put those amino acids together in order to put them in the right shape? It all comes back to the genes. Remember, a gene is a sequence of DNA, it's a sequence of instructions, and that sequence of instructions dictates how the protein is supposed to be made. So that means then, in order to build proteins, you first have to start with what? Well, you first have to start with DNA, and then from there, you have the instructions that tell us what to build. But DNA is not the only nucleic acid that's used in this process. This process is also going to involve the nucleic acid known as RNA. Now, if you recall, RNA stands for ribonucleic acid, and there's a few differences that exist between DNA and RNA. The first big difference is the structure itself. Remember, DNA is a double helix. It's double-stranded. RNA, on the other hand, is a single strand. Now, because DNA and RNA are both nucleic acids, they're made of the same things. They're made of phosphate sugars and bases. But if you recall, DNA is made of the sugar deoxyribose, where RNA is made of the sugar ribose. They both are made of phosphate groups, and then they're both made of nitrogenous bases. But again, if you recall, the four bases that exist in DNA are adenine, thymine, guanine, and cytosine. So you remember Auburn Tigers, A went with T. Well, in RNA, there is no T. There is no thymine. Instead, RNA possesses the four bases adenine, uracil, guanine, and cytosine. So when RNA bases pair, adenine pairs with uracil, guanine still with cytosine. So as we just discussed, RNA is like DNA in that it's a nucleic acid, therefore it's made of nucleotides. But what I have a lot of people ask is that why do you even need RNA here? If DNA carries the instructions, why is RNA needed? And it all comes back to the structure. Remember, DNA is a double helix found inside of the nucleus of a cell. RNA is single-stranded. So the problem with DNA is too big to actually get outside of that nucleus. So what happens is we take the DNA that carries our instructions, we use it as a template to make RNA, which just happens to be small enough it can leave the nucleus, and it can carry those instructions out into the cytoplasm where you find ribosomes. Now, why are ribosomes involved? Well, remember, it's ribosomes that are the protein factories. So again, why can't DNA do all this by itself? Because DNA is too big to leave the nucleus. So we're going to take DNA, use it to make RNA, and then RNA can be used to actually build the proteins. So we have a couple of names for the flow of genetic information through this protein synthesis. So two steps here, transcription and translation. Transcription is the first half of protein synthesis where we start with the DNA and we use it to make RNA. And then translation follows where we take RNA and use it to build the protein. Now if you look at the image on the bottom of the screen, this is the image from page 179 in your text, you'll notice here we are with our double-stranded DNA. Now because this is DNA, all the A's are with T's and the G's are with C's. But remember, this is too large to leave the nucleus, so we want to take this double helix DNA, we're going to use it as a guide to build RNA. Now in building RNA, and we're going to talk this process in more detail in just a second, but basically your rule changes. So C's are still with G's, G's are still with C's. T is still with A here because A can be present in RNA. But if there's an A in DNA, it cannot go with T because RNA does not have a T. Instead, it pairs with U here. And then translation takes that RNA and uses it to build a protein by telling us which amino acids we need in which order. Because remember, the order we put amino acids together in, it folds that protein into the correct shape. So let's start with the first process in this section, and then we'll come back to translation but later on. So again, transcription, we're taking DNA and we're using it to make RNA. So essentially what we're doing is we're copying DNA into RNA bases. It's the same way that a transcript of a speech is written down. So if you had like an oral presentation and then you've got somebody off to the side that's writing out everything that's said. So it's just copying the same information, but transcription here, as it copies the same information, it puts it into a sequence of RNA bases versus DNA because, again, remember, it's a smaller size. Now, because DNA is involved, this process happens entirely inside of the nucleus because, remember, DNA can't get out. And then there's a really important structure that's used in transcription. It's known as RNA polymerase. Now, before we go any further, what is RNA polymerase? Well, because of its ending ASE, you automatically know it's an enzyme. So what does this enzyme do? Well, this enzyme binds to every nucleotide sequence at the beginning of every gene, which is known as the promoter, so where this whole process starts. So the RNA polymerase is going to bind to the DNA. It's going to unzip the double helix. Now, unzip is just another word here for separating the double helix in half. And then once it has split the DNA in half, it matches RNA nucleotides with complementary DNA nucleotides. And the result is a single-stranded messenger RNA that has the job of carrying the message, which is the instructions, to build the protein. Now, there's a really good image here. I'm not going to let you listen to it, but I did just want to show you. So here, this green structure in the back, this is the RNA polymerase, and of course, this is our DNA double helix. So the first thing polymerase does is it unzips, or it unwinds, or it separates the double helix in half. And then once it has separated it, you'll notice it's going to find RNA nucleotides that are out in the cell, and it's going to bind them to the DNA nucleotides. So you'll notice all the the thymines that are in DNA bind with adenines in RNA. The adenines in DNA bind with uracil. G's are still with C's, C's with G's. And so once it pairs the RNA bases up with DNA, then eventually the RNA is going to be released, and it's now small enough it can leave the nucleus and carry the message out. Now, how does it carry the same message? Because remember, these were paired in the same order as the DNA bases. So it's carrying the instructions just in a smaller format that can actually leave the nucleus and go out to build the protein.